Hello, everyone. My name is Mina Rowe Johnson, and I'm a faculty member in the Department of Biochemistry. And I'll be going over some of the content that we'll be covering in the first class of the cell biology module, with the theme being that cellular processes are dynamic. And so when you think about how you might infer a function in a cell for a particular protein or a complex of proteins in a cell, you might be thinking about, well, what if I were to look at the localization of those proteins in the cell? That will probably tell me what that protein is doing in that cell for normal behavior. And it's kind of akin to Google Maps. If you were to look at a map of sort of part of Salt Lake City, you might see something like this. So this area here, there are a lot of compactors, grapplers, metal shears. And you might say, okay, based on the localization of these components, the function of this spot, this location, might be recycling or some sort of manufacturing. And you would be right, because this is the sort of the recycling depot. What about the case here where all of these little like things that look like Tic Tacs are school buses? And so you might think, oh, the school buses must function here, right? But actually what it is, it's a school bus depot. And you're looking at a map maybe at around, I don't know, 2 o'clock, maybe 1 o'clock, when the schools are still in session and the school buses aren't really being used. And so they're all parked here at this school bus depot. But if you were to look at 9 in the morning or 3 p.m. when school starts and school gets out, what you'll see are these school buses sort of all along the roads, because their function really isn't at this school depot, right? If you look over time, you'll see that their function is to move from schools to other locations. And so that's what we mean by sort of what is cell biology. It's a coordination of all the things in the city that make the city run. And the fact that many of the processes in a city or in a cell are dynamic. And so if we were to use this analogy a little bit further, genetics, which you've gone over in the earlier part of foundations, are really sort of the building blocks, right? So in our case of the city, they might be the components that make up the city, like the individual buses, or maybe the bus seats within the buses, the roads, the schools. And then you talked about protein-protein interactions in the middle of the module. And that might be, maybe some schools, because of their enrollment, they actually need two buses to come there, and the interaction between buses to coordinate that response to get the kids from, location 1 to location 2. And cell biology really are how these processes are then integrated. So you have buses and you have schools, but in order to link these two concepts, you need streets and traffic lights, and the buses sort of respond to the traffic lights along these streets to get to the schools or to the locations they need to get to. And of course, all of these things don't really act in isolation, but they're integrated with one another. It's not so easy actually to draw a clear line between, what specifically is protein interactions versus what is cell biology. And it's really the dynamic interplay between these concepts that allow a cell to function. And so if any of these processes go awry, let's say, we lose power and all the traffic lights stop working, and you get this pileup, it affects not just that location, but really the city as a whole. You know as well as I do when there's construction on campus that it really messes up traffic, really all the way down to Foothill. So it requires sort of the precise control of all of these dynamic processes in order to have the entire cell to work properly and to respond. So as you see here, in response to this many car pileup, there are emergency vehicles. So cells also have processes in place to be able to correct these sort of failures or lack of control. So you might be thinking, I really like diseases, and so I don't particularly care about cell biology or fundamental aspects of cell behavior. So I'd like for you to take a moment and think about what kind of human diseases, maybe the human disease that you're really interested in, can be attributed to dysregulation of a fundamental process that a cell does. And as you think about those examples, be thinking about, well, what are sort of the root causes for some of the disease phenotypes that I'm interested in. And I think what you'll probably find if you take a moment and think about it is it's really everything. And I've just listed a few examples here that might be of interest to the students in the class, but it's almost all human diseases can be attributed to dysregulation of either genetics or cell biological processes. And so understanding fundamental aspects of cell behavior is key to understanding root causes of human diseases. Okay, so again, the theme of today's, of the first class will be cellular processes are dynamic. And so here's this one image of this picture at one point in time. So you might think this is obviously a street and the cars are driving along it, but honestly, you don't really know because it's a static image. What if I were to say, actually, it's a parking strip and these cells are not moving at all? You wouldn't know unless you had either a time frame before it or a time frame after it to be able to understand whether whether these cars are moving or not. Perhaps you can infer based on the fact that, there's clearly a yellow line in the middle, and there's sort of dotted lines, and it looks kind of like a highway, so they're probably not parked. But the point being is that a lot of the processes that we're interested in looking at in the cell require looking at things over time. And so today we'll be talking about the cytoskeleton as an example of a cellular process in the cell that is incredibly dynamic. So this is an example of a cell where the three components of the cytoskeleton, actin, microtubules, and intermediate filaments, are labeled in different colors. And you can see that the cytoskeleton of the cell really is, as its name applies, it creates the skeleton of the cell. It gives the cell its structure. It also acts as highways for different cargoes to move around. And it is tightly coordinated with other cellular processes for homeostasis. So actin and microtubules are extremely dynamic. We're going to go a little bit more into that in this slide deck. Intermediate filaments are also a cytoskeletal component. They're a little less dynamic, but I think it's getting a lot more attention in terms of they're much more dynamic than we thought that they were, but they don't have the same sort of regulation that actin and microtubules have. Okay. So when I say dynamic but stable, what I mean is you kind of need the skeleton to be this sort of structurally sound component of the cell because it will give it shape. But the actual filaments that you observe here are not static filaments, that those are dynamic in itself. So why would you need to change a cell shape? Well, here's an example, a beautiful example. It's an oldie but a goodie of a leukocyte that's chasing down this bacterium. So here's this bacterium wobbling around, and this leukocyte is responding to cues from this bacterium and trying to get at it. And actually, the movie stops right before the best part, which is when the leukocyte eats the bacterium and then just stops moving because it's satiated. And these little dark gray things are red blood cells that the leukocyte is sort of pushing out of the way. So for a cell to move like this, you need the cytoskeleton, the skeleton of that cell, to remodel, right? You need the front to push out. You need that to be coordinated with the back pulling in. And it's just this constant cycle of pushing and contracting for this cell to move. So when you think about what are the cytoskeletal components that are required for this cell to change shape during cell migration, one of the main components, actually all three are very much involved, but one of the examples I'll be going over with you is actin. So actin is a filament. You sort of saw that in this image here. So actin is the blue, and you can kind of see them as like threads, long structures of blue that are sort of running through the cell and kind of at the periphery of the cell too. But these actin filaments are actually comprised up of actin monomers. And so actin monomers themselves have a polarity associated to them. And what I mean by polarity is there's like a top and a bottom to that monomer that we refer to as the plus end or the minus end. And then the actin monomers sort of assemble along there. So it's like minus N plus N, and then another minus N plus N, another minus N plus N. So these monomers sort of stack on top of one another to form this filament. You can see that it's sort of two filaments that are twisted, and that gives the structure some structural integrity. If it was just this long straight filament, you could imagine it like kind of like a spaghetti noodle, a cooked spaghetti noodle, where it would bend under pressure. And that means that since the monomers have a polarity associated with it, the filaments also have a polarity associated. So the plus end, which we sometimes refer to as sort of the business end, and I'll go into why, is at one end of that actin filament is also called the barbed end. And then the opposite end is called the minus end, or also referred to as the pointed end. And you can see them here also with electron microscopy at the ultrastructural level. You can even make out the tiny little like bulbous structures, which are these actin monomers within this actin filament. So where is actin? Well, in the image that I showed you before, it's sort of everywhere. But when you think about when a cell needs to move, so this is a side view of a cell sort of migrating along a surface that looks kind of like that, right? So when the cell is moving, it needs to sort of push out a front. This is obviously like kind of dramatic here, but you get this protrusion to the front that's called the lamellipodium. It then needs to stick down such that it can kind of grip on to the bottom, so that when the cell pulls itself forward, it can contract from the back and then get rid of the adhesions from the back. And then you see this like net cell body translocation forward. So the forces required to push out that plasma membrane to make this structure, this protrusion, this lamellipodium, is mainly actin mediated. So now if you do like a top view, looking down on a cell, migrating on a dish, all the white stuff here is the actin localization in that cell. And you can see that there's rich actin dense work right at the front of the cell where it needs to push out. So it would be like equivalent to this part right here. And of course, it's not that the actin is only there. There are these actin structures that are sort of more proximal, like closer to the cell body, that still maintain the shape of the cell as it pushes out this actin-rich protrusion in the front. So I told you that actin monomers make actin filaments, and that there's lots of actin sort of at the front of the cell where it needs to push out against the membrane to form this protrusion. So how is this all coordinated? So again, we're looking at the top view of a cell. So it's kind of like looking at this view here, except for now the protrusion is sort of at the top. So if you look at the plasma membrane, you have it sort of flat first, and then it starts to bend out, and it starts to bend out even more here as the cell is in this cartoon is beginning to make a protrusion. And these blue things are the actin filaments. So it's not just, actin filaments working in isolation. They start to form this branched network. And this sort of coordination for these filaments to form these branched networks with the plus ends or the barbed ends at the right near the plasma membrane allows additional monomers to be added on to those growing ends of the actin filament to begin pushing the membrane forward. And that is how actin filaments assemble to be able to cause the forces to result in the forces required to make a protrusion. Sometimes the protrusion takes, quite a bit of force. In that case, the actin will begin to bundle. So not just forming this network, but it'll form these like stick-like structures, and the cell shape will reflect that. It'll form more like a finger-like protrusion versus more of this flat, broad protrusion. So the more branched it is, it's broad, but it still pushes out. The more bundled it is, you get these like stick-like things called filopodia. Again, the shape of the cell is dictated by its cytoskeleton. And the cytoskeleton is a dynamic structure that grows and shrinks to result in these shape changes. So growing and shrinking, when you look at an actin filament, it is constantly undergoing treadmilling. So what treadmilling is kind of, when you're on a treadmill and you're running, you're not really going anywhere, but the thing under you keeps moving, right? So what this is if you look at an actin filament, let's say at time point 0, and then you look at the actin filament at some point later, it's the same length. But that doesn't mean that it's just this static structure. If I label part of that actin filament in red, what we would see is that part slowly tracking backward to the minus end over time. And that is through a process called treadmilling. So in this case, labeled actin is being assembled on the plus end at the same rate as it's being disassembled from the minus end. So it's constantly going assembly and disassembly. It's just that since the rates are equal, the length of that filament stays the same over time. And that is almost all actin filaments in the cell are undergoing treadmilling. Same principle happens with microtubules, where they're constantly undergoing flux, right? Adding and taking away, but that the rates are the same. When you want growth, though, filament growth, what you'll begin to see is that the rate of assembly is just faster than the rate of disassembly. So you'll have this net increase, right, in filament length over time, but it doesn't mean that this end just stops moving or like nothing happens back there. It's that you just get the faster rate of polymerization at this plus end. And then a lot of the rates of polymerization and depolymerization are mediated by two things. First, the ATP status of the actin molecule monomer. So actin is ATP loaded, and it begins to undergo hydrolysis as it stays associated with the filament. And it's actually that ATP hydrolysis that makes the monomer sort of lose affinity for the filament and then come off. So this red part is actually supposed to, well, in this case, it's labeled actin, but you'll see later, you'll see the ATP-ADP cycles. Okay, so actin is constantly undergoing treadmilling, but it can stay a constant length with this treadmilling, but it can also grow if the assembly rate is faster than the disassembly rate. And if you want more information on ATP status, as well as what drives the assembly and disassembly rates, it also is related to the actin monomer concentration in the local area. You can get more information on panel 16.2, which is on page 960, 961. I found that these two pages are really helpful to get kind of a bird's eye view on actin and microtubule dynamics. Okay, so I'm going to exit out and show you this really great video that I found. And I'll narrate it as it goes. And it kind of goes over the cycles of sort of actin assembly and disassembly. So here we have an actin monomer. Shows you sort of the crystal structure of the actin monomer. ATP hydrolysis is the energy required for filament assembly and disassembly. So this actin monomer is now ATP loaded. It's showing you an example of ATP hydrolysis. So now you have ADP, actin. So you get this, actin monomers are sort of floating around. You need 3 actin monomers to come together to start assembling the filament. That's called spontaneous nucleation. So here we have 3 actin monomers coming together, and now the filament starts to assemble. Remember, it has a polarity, a minus end and a plus end. You get a lot of growth at the plus end. This blue to yellow shows ATP hydrolysis, and now it's starting to kind of break off, right? The yellow guys are ATP loaded. These ones are ADP. Now, when all of the actin is ADP, you start to get depolymerization of the actin filament. I'm going to pause there because we don't need to learn about all the actin accessory proteins. So I hope that kind of goes over for you the dynamic processes of an actin filament. But remember, actin isn't the sole player of the cytoskeleton. There are 2 other components of the cytoskeleton, microtubules and intermediate filaments. And at least for microtubules, shown here in green, the same principles apply, although there are obviously some differences between actin and microtubules. But it's this idea that microtubule dimers that's shown here, again, go through hydrolysis, nucleotide hydrolysis. In this case, it's GTP to GDP. And these tubulin dimers come together and assemble this protofilament. So these dimers sort of coil around each other like this and form this 18 units sort of hollow tube. Again, if it was just a dimer forming a filament by itself, just this long, thin structure, it just would not have the structural integrity to meet the demands of the forces that the cell experiences. So by forming these tubes, it gives it structural integrity. So you see this GTP-loaded tubulin dimers, again, assembled to the plus end of the microtubule, just like actin. Microtubule also has a polarity. And as it gets assembled and starts moving down in a process called treadmilling, the GTP begins to get hydrolyzed and is GDP bound, and then the GDP tubulin starts to lose affinity with the tube and begins to disassemble. But sometimes the GDP tubulin catches up with the plus end. And what you get is this spontaneous disassembly called catastrophe, because it's sort of this catastrophic event where the plus end of the microtubule completely splits and just shrinks all the way down. It's incredibly fast. It's actually a really great feature of microtubules because their dynamics are actually quite a bit faster than actin filaments. And then you start to see the cycle kind of return where you get more GTP loaded tubulin growing again on the plus end. So what this example doesn't show, but happens, and actually the cartoon that we just saw on YouTube didn't show it well too, is it seems like all the business happens here at the plus end, but that's not true. You can get dimer addition on the plus end, but you could also get dimer loss on the minus end, and sometimes you even get lateral incorporation of dimers. So there are quite a few ways that you could contribute sort of to the growth and disassembly of microtubule dimers, and that's also true for actin. So why would this matter? Why would it be important for microtubules to be able to shrink really quickly and then grow and shrink really quickly? Well, as many of you may know, microtubules drive a really key part of the cellular process called cell division. And that's the division of the chromosomes into two separate daughter cells. So shown here is an example of a cell at metaphase. That's when the chromosomes are all aligned at the middle of the cell. And these microtubules shown here in red are attaching onto the chromosomes and then are going to split them apart. So you need this microtubule to shrink very quickly, right, in order to pull these sister chromatids apart. And that happens through the regulation of microtubules at that plus end depolymerizing. So here's an example of that. So in red are microtubules. Green are the areas where the microtubules are making attachments with the chromosomes, and they're it's called a kinetochore. And these black sort of sausagey-like things are the actual chromosomes, which are unlabeled. When I play this movie, what you'll see is even at the spot of metaphase, where you think it's just this still structure where the chromosomes are just kind of staying still, it's incredibly dynamic. There's this push and pull, this sort of like tug of war on both sides. And actually, this is a really important part for the fidelity of cell division, which I won't go into, but it's incredibly rapid. So the microtubules have to be dynamic for this process to occur reliably. So for a process that's this dynamic, if let's say you're putting together a paper and you wanted to show, man, these structures are just super dynamic. How am I going to show that in 2D space in print, right? You can put movies up, but there are figures in a paper too. And so how would you show that? Well, the way that we do this in cell biology is through a chymograph. Okay, what this is if I took a line, so I'm going to pause right here. If I took a line right through one of these pairs of sister kinetochores here, like that, and I plotted that line right here. And then I plotted that same one over time as the movie plays. So time is now on the y-axis. What you'll see is those two kinetochores going woo like this, right? Back and forth, back and forth. And that's actually reflected in the chymograph, which you also see in the movie, right? Back and forth, back and forth, back and forth. And so a chymograph is a really great way to show a process that's dynamic, but you have to show that sort of in two-dimensional space, like in a static image. And if this is confusing for you, I don't think the textbook goes into chymographs. You could try Googling chymographs, but I'm also happy to go over this in class further, and we'll talk more about it in class too. So because these are all dynamic processes, microtubules as well as actin, there are inhibitors that have been made against these processes, either at those plus ends or those minus ends, or to stabilize the filament. So these inhibitors have been great for functional studies for actin and microtubules, and for a list of them and how they function, you can look at table 16.1 on page 964. Okay, so today I wanted to go over how cellular processes are dynamic, and how that's required for the cell to function, or the city to function in this case too. But I only talked about actin and microtubules. There are many, many more processes in the cell that are dynamic for homeostasis. So as an example, if we go back to this Google Maps, if we look at a bus making stops, right, it usually just doesn't go from like one spot to another. It needs to take defined paths. If it wants to go from stop 1 to stop 2, it can't just take like a straight line because it'll go right through all of these buildings. There are rules that it needs to follow, mostly streets and street signs that it needs to follow to get to 2. And then maybe after that, it wants to go onto the highway and it can't just jump over here. It needs to go onto an on-ramp. So again, this analogy is similar to something like vesicles carrying cargo, right? There are rules that these vesicles need to follow. They often march along filaments. There are adapter proteins that link that vesicle onto a specific filament and going in a specific direction, just like some of our streets are one way. You can't even go, both directions. And so just want to leave you with this idea that even though the cell is under homeostasis, as in everything is sort of working as it should be, that process itself is incredibly dynamic. Okay, so I look forward to seeing you in class.